Yes, I'm going to give the briefest introduction for Cyrillus. So Cyrillus came to the Bioinformatics program from Emory, where he did his dual degrees in biology and computer science. And I think something that Cyrillus will show us in a bit is that he has pretty diverse interests, but one of the common themes from that is his interest in how mathematics can tell us about the world around us. And that makes it a lot of fun to work with Cyrillus because he's interested in how the ideas of mathematical biology that we're exploring can also connect to surprising other areas of science and of other areas of study. So with that, Cyrillus, take it away.
Well, thank you, Dr. Meyer, for the introduction. So I'm Cyrillus, as he said, and I want to start with this talk with something more personal and fun, isn't it? So as Dr. Meyer said, I graduated from Emory University in the year of 2018. So before I went to college, I have a lot of struggle of deciding what I want to pursue as my career. My favorite subject science subject in high school was chemistry, due to have a very good teacher at that time. And when I applied to college, I wrote a whole essay about how I'm so passionate about neuroscience and psychology and what I want to do. But eventually, when I come to college, I realized the curriculum is somehow different from what I thought. So I slowly shifted to biology. Another influence from high school is my high school counselor told me that my personality will be pretty well fit into social science. And the crown of social science, of course, is economics. So actually, the first major I declared in college was economics. And I spent a decent amount of time on that before I decided to switch into computer science. But I was one step away from getting a degree in economics. So it's kind of go to waste right now. But I think one good influence of study social science is give me a lot of perspective of how we should think about our society. And I also have a chance to get to know a lot of big figures in the giants of how to think about our society. One of them is Judge Learned Hand. He has said one of my favorite quotes that I want to share with you today, which is from his speech after World War II. He said, "The spirit of liberty is the spirit which is not too sure that it is right." I think this is a very good advice to give to everyone who's working the vocation of science, as well as any concerned citizens who seek the way to bring our society together. Well, the reason I bring up Judge Hand today, on the one hand, pun intended, is of course to introduce you to my scientific journey and my intellectual curiosities. But on the other hand, I want to really take this as an introduction of what I want to talk about today. So one of the key contributions for Judge Hand is he brings math and economics into the field of law. For example, if you want to debate in the court, say that someone has breached his duty of care and caused you harm, what would you say, right? Of course, we as a society, we have to make a balance. We cannot spend limitless amount of resource onto something that is very unlikely to happen. But on the other side, we should take enough precaution to prevent people from getting harm. So what Judge Hand said is really he think about this question as in a math formula, which is given here. I think my audience today have no problems understanding what is the logic behind this formula. And it's from the economics perspective, it's really a cost utility function. But with this function, he very clearly lined out what we should consider when we think about these cases. The interesting thing is if you are a law student, you will never use this formula explicitly when you write an essay in your exam or debate in front of a court. But what is important is the thought process embedded in this formula. So the point I try to make is sometimes when we have a mathematical equation or build a math model, it's not really about the specific prediction it has, just like no one would grab a calculator in front of a judge and calculate the likelihood of something happened. And also, this simple formula cannot cover the thousands of different cases in tort. But this mathematical model can provide with us a very good general guidance on how we should approach questions like this and what are the factors we should consider. Which bring me to my conversation today in biology. My talk today is something very different about drug design. And So here I give some very interesting examples, current advances in this current decade about the cytokine therapy, about the CAR T-cell therapy, and the engineering in antibodies, such as bispecific or even trispecific antibodies. If there's something common about these drugs and many other drugs you can think of, it's really about the fact that we want them to bind to the cell that we care about. Just like the whole effort we go through in the CAR T-cell therapy is to make sure that they bind to the cancer cells instead of our normal cells. So I think the key word here is selective. I steal this figure from a review journal that talk about how if we bind to the intended tissue and intended receptors, our drug can give us very good effect, versus if we have any binding to off-target cells and off-target receptors, will lead to reduced effectiveness, and sometimes will even lead to unintended effects, side effects. So the whole idea here is, how can we design drugs that can bind to our intended tissue selectively? So of course, that is impossible for me to answer this question in this 20 minutes talk. But as I said, if we have a mathematical model and have a very quantitative view, we can make some very interesting argument as a general guideline. So which is what I'm going to do today. To have a mathematical model, first we have to make some theoretical assumptions. So the first one is, how do we define cell populations, right? If you are in the field of biology, you probably are pretty familiar with this plot, the flow cytometry chart. And in the flow cytometry setting, we get to know that this can measure the amount of receptors or surface protein expressed by some cells. And we plot them in the 2D space of receptor 1 and 2. And each dot on this plot, of course, is one cell. And through some gating, we can separate one cell population from the other. So the story here is the cell population is really defined in biology by the receptors they express. So with this spirit, we come up with the theoretical case that, let's say we have two theoretical receptors, R1 and R2, and plot it right over here. And we can draw some, arbitrarily draw some cell populations on this 2D space. And these are our cell populations. If you have some hard time visualizing what's going on here, we have some creepy cartoons here for you. But the idea here is with these definitions of cell populations, we can start to define, or we can start to call, which cell populations are the target one and which one are the off-target one for some more theoretical analysis. The second question here is, how do we know how much binding we have on each cell? So here introduce our binding model. We developed this mechanistic binding model, which is not too much magic other than the law of mass action. But I think the innovation here is it allows multiple receptors and many ligands to interact and have this binding process, a multivalent binding process. So this binding model basically take a binding affinity between ligands, each pair of ligands and receptors as parameters, and also the level of receptor expression on the cell, and give out the prediction of how much binding we'll have during equilibrium state. So once we have these two pieces together, we can bring them in under one picture. For each cell, we can plot them in this 2D space, and for receptor 1 and 2. And to plug them into our binding model, we can make a prediction, and we will plot them with a color. And this will make this heat map, and we'll also draw the contours on that. And this 2D space is no different from the space that we formulate those 8 theoretical cell populations. So we can really overlap what's happening here. And it's very easy for us to read out how much binding we have to read from the contour plot. Well, one thing we care about in this project is not really about the absolute amount of binding happening on each cell population, but the selectivity, right? The relative amount of binding. So here we define the selectivity as the amount of binding on the target population versus divided by the amount of binding on off-target populations. And any kind of engineering strategy we're trying to do here is to optimize the selectivity. And one good thing about this plot, of course, is you can read out the amount of binding as well as selectivity from this plot by seeing how many contour how many contour lines we have between two populations. With the theoretical setup, I think now I can show you that we explore several engineering strategies with this framework. So the first strategy I want to talk about is affinity modulation. Affinity modulation is a very straightforward one, that if we can do some protein engineering in our drug and modify the binding affinity to some of the receptors. So to investigate this issue, what we do is we plot out the binding contour plot for many cases with the variation of the binding affinity to the two receptors varying 100-fold. And when we have these plots, we can read out the selectivity from comparing each pair of populations. And what we find out is that this affinity modulation is really the most effective when our target and off-target populations have very divergently expressed receptors. For example, in this case, when our target receptors, target cell populations have very high receptor 1, but our off-target have very high receptor level of 2, then what we can do very intuitively, we can enhance the drug's binding affinity to receptor 1, but to diminish the binding affinity to receptor 2 to achieve the optimal selectivity. The second strategy is the valency modulation. While many drugs exist in this monovalent form, the monomer form, we can also make a polymer out of this drug and make them together. For example, this is a tetravalent drug. And we want to investigate if this approach will help us to increase or to optimize the selectivity. As the heat contour map is showing here, that when the valency is increasing from 1 to 4 to 16, it really exhibits a nonlinear increase in how much amount of binding it has. Of course, we'll care more about the selectivity, so we'll plot them out. As we predicted here too, that for any specific drug, the effect of valency on selectivity really have this non-monotonic trend, and there is a optimal valency somehow between the lowest and the highest ones. And even further, we investigate here when we compare these two populations, is that the effect of valency is also dependent on the affinity of the monomers. So we plot out the low, medium, and high affinity, and our discovery is the best case usually is when we have a multivalent drug, but the binding affinity of each monomer is relatively low. And these cases usually give the best selectivity. We have to do some literature research on this topic, and there are several experimental work actually validate this discovery that we predicted in our model. The third strategy I want to talk about today is a mixture. So what a mixture is about is, as the diagram shows here, let's say we have 30% of drug 1 and 70% of drug 2, and we wonder if this cocktail of drug can bring any better selectivity than using one drug by itself. So when we compare several population pairs, we realize that the effect of mixture is really additive, which means using one drug is using the combination of drug is really no better than using one single drug by itself because the trend is monotonic. Well, there are some modest improvement if we have more than two or more than two off-target populations when these two populations have very divergent expression profiles, such as this case. When we have singly high R1 and singly high R2, then using the mixture can bring some very modest improvement in the mixture case. The fourth strategy is bispecificity. So previously I've talked about bivalent drug, which is the drug of valency 2. And bispecificity is basically a special case of that, which is a bivalent drug, but we want different subunit have to be different. So here, of course, we want to seek some unique advantages of using that compared to homogeneous bivalent drug. But when we plot out this, we realize that if we only care about the amount of ligand bind, this behavior is not too much different from bivalent behavior. But luckily, when many bispecific drugs are used in real life, that requires both sides to be bounded to be effective. For example, one very good example is the bispecific antibodies. What do I mean by bind to both sides? So previously, we only care about if there's any binding. If there's just one one subunit bind to receptor, we will count that as bind. But in this case, we will care about if we only count one ligand if both of their subunits are bind to receptors. And we find out that this fully bound case do show some very interesting characteristic. Specifically, that the bispecific drug will prefer doubly positive cell populations compared to single positive cell populations. And there are many efforts in the pharmaceutical industry that want to take advantage of these characteristics. There are some other strategies we have tried out in our work that I don't have time to talk about today, but I will just briefly introduce. We have to think about if we can combine the 4 strategies I've talked about, and if this combination can really bring some synergistics effect among the strategies. And we will find out that there is some synergistic effect among the strategies that we can optimize. Another thing we'll try out is if we can introduce some binding competition, which is very common in biology, that we introduce an antagonist or a dead ligand that doesn't do something itself, but it changes the binding behavior of other drugs. And we do find out that introducing this dead ligand will invert the receptor targeting, at least in this contour map, which is a very interesting characteristic we might be able to take advantage of. To zoom out to the larger picture about drug design in general, the whole history of human designing drugs is really to find out the more specific molecular target, right? From 50 years ago, when we have this revolution of rational drug discovery, we really try to specify what kind of molecule, what kind of receptors, and what kind of cells that our drugs are targeting. And now in the year of 2021, there are many efforts as invested in how to design multispecific drug. Of course, the challenge in multispecific drug is the vast probability that we can do with combining things together. And there's also the complexity of the combinatorial large amount of binding configuration that can happen. So this new wave, this new horizon of drug design really brings a new potential of having a mathematical binding model and provide with us even better guidelines of how we do these things. So one ongoing effort of mine is to formulate these in a more mathematical way, formulate this binding model in a more mathematical way, so more people can take advantage of this math model. And we also offer to make this a package so more people can play with it and make more discoveries. So as a summary today, I think I somehow started talking about Judge Hand, but the whole story is just sweeping to biology, and I think you guys don't mind that. But the point I try to make is having a mathematical model and a computational model can really help us to provide some very general guideline and make some very good insight on the topic of interest. And also use this multivalent binding model to make argument that strategies such as affinity modulation, valency mixture, and complex engineering can enhance the selectivity of cell binding in each specific situation. So with that, I want to conclude my talk today. I would like to thank my advisor, Dr. Meyer, and my homie Brian for working with me on this project. I have always been the spokesman of this project, but this is really a group effort. So I would like to invite you to get to know people behind the scene. And Dr. Meyer, I think you guys already see Dr. Meyer here. Brian, do you want to come out and take a bow?
Thanks for the shout out, Cyrillus. It's been great working with you.
And we have been finishing this manuscript and post on bioRxiv. So if you want to check on this, this is the link. So with that, I want to thank you for your attention.
I have a question. Hi, Cyrillus. So that was super interesting, and I'm going to read your paper. But so I was thinking, like, a lot of what you're looking at is more on the ligand side of things, to see like how the ligands, like maybe how much of a polymer they are, or the dosage on that from that lens. I'm curious if you looked at, let's say, heterogeneity on the cell type side, like maybe they have a different proportion of receptors on their cells, and if you can kind of use that angle to do some sort of like personalized medicine, where if you like drew somebody's blood, you could see what what their cells kind of look like, and then build the dosage based on that.
Yeah. So in this binding model, of course, we have it's embedded that we consider the amount of receptors on cells, right? So this model fully have the capacity of working with different amount of receptors. Of course, in the work I'm showing you today, we don't really focus on that as much, but that is easily doable with the spiny model.
I just wanted to chime in. This is really nice work, Cyrillus. I really enjoyed your talk. I was just curious, in our own field, in neuroscience, in experimental neuroscience, not necessarily the the human disease side of things. There is all these designer drugs that are coming along where you try to sort of target specific cell types and specific receptors on those cell types and things like that. What is your thought on how we could exploit something like this in that area?
Well, first of all, you have very good talk in neuroscience, and thank you for the compliment. I think I'm not like very knowledgeable in that case, although I claim to be used to be a neuroscience major, that I didn't really pursue that part. But I think there are difficult parts of applying that to the highly complicated tissues, such as neurons, right? Because it's not just about, in this case, we only care about binding, and there's a lot of things this model doesn't consider. For example, the connectivity between different neurons. That's one thing, right? And there are many molecular mechanisms that inside one cells about how to regulate the receptors. So of course, I'm not saying this model can do all these things. But do you have any specifics, drugs you want to talk about in the neuroscience?
I don't have anything specific in mind. I was just curious because people are coming up with designer drugs that activate specific neurons, and it's in an experimental setting, not from a therapeutic standpoint. I was just curious how these more generic models can motivate or even advance designing those types of drugs and receptors as well. In fact, those are designer receptors, which makes it even more exciting because you can express those receptors on whichever cell type you want. And this way you can make up a ligand and receptor combination with known binding properties. And that could actually be leveraged quite well if one were to think of something like that.
Yeah, absolutely. I think there's at least some way I could come up with. You talk about personalized medicine, and the expression level of different receptors can be different from people to people. So that's something we can definitely take advantage of. And the second thing is the subpopulation of different cells also have different expressions. And that the subtle differences is also something we really can take advantage of through a more quantitative perspective, right?
Very good work, very broadly applicable, very nice.
Thank you.